T-cell receptors are antibody-like heterodimers. Because T-cell responses depend on direct contact with an antigen-presenting cell or a target cell, the antigen receptors made by T-cells, unlike antibodies made by B-cells, exist only in membrane-bound form and are not secreted. For this reason, T-cell receptors were difficult to isolate, and it was not until the 1980s that they were first identified biochemically. On both cytotoxic and helper T-cells, the receptors are similar to antibodies. They are composed of two disulfide linked polypeptide chains called alpha and beta, each of which contains two Ig-like domains, one variable and one constant. Moreover, the three-dimensional structure of the extracellular part of a T-cell receptor has been determined by X-ray diffraction, and it looks very much like one arm of a Y-shaped antibody molecule. A T-cell receptor heterodimer. Schematic drawing showing that the receptor is composed of an alpha and beta polypeptide chain. Each chain is about 280 amino acids long and has a large extracellular part that is folded into two Ig-like domains. The pools of gene segments that encode the alpha and beta chains are located on different chromosomes. Like antibody heavy chain pools, the T-cell receptor pools contain separate V, D, and J gene segments which are brought together by site-specific recombination during T-cell development in the thymus. With one exception, all the mechanisms used by B cells to generate antibody diversity are also used by T cells to generate T-cell receptor diversity. Indeed, some VDJ recombinase is used, including the RAG proteins discussed earlier. The mechanism that does not operate in T-cell receptor diversification is antigen-driven somatic hypermutation. Thus, the affinity of the receptors remain low, Ka 10 to the 5 to 10 to the 7 liters per mole. Even late in an immune response, we discussed later how various co-receptors and cell-to-cell adhesion mechanisms greatly strengthen the binding of a T cell to an antigen-presenting cell or a target cell, helping to compensate for the low affinity of the T cell receptors. A small minority of T cells, instead of making alpha and beta chains, make a different but related type of receptor heterodimer composed of gamma and delta chains. These cells arise early in development and are found mainly in the epithelia, in the skin and gut, for example. Their functions are uncertain, and we shall not discuss them further. As with antigen receptors on B cells, the T cell receptors are tightly associated in the plasma membrane with a number of invariant membrane-bound proteins that are involved in passing the signal from an antigen-activated receptor to the cell interior. We discuss these proteins in more detail later. First, however, we need to consider how cytotoxic and helper T cells function and the special ways in which they recognize foreign antigen. Antigen-presenting cells activate T cells. Before cytotoxic or helper T cells can kill or help their target cells, respectively, they must be activated to proliferate and differentiate into effector cells. This activation occurs in peripheral lymphoid organs on the surface of antigen-presenting cells that display foreign antigen complex with MHC proteins on their surface. There are three main types of antigen-presenting cells in peripheral lymphoid organs that can activate T cells: dendritic cells, macrophages, and B cells. The most potent of these are dendritic cells, whose only known function is to present foreign antigens to T cells. Immature dendritic cells are located in tissues throughout the body, including the skin, gut, and respiratory tract. When they encounter invading microbes at these sites, they endocytose the pathogens of their products and carry them via the lymph to local lymph nodes or gut-associated lymphoid organs. The encounter with pathogen includes dendritic cell to mature from an antigen-capturing cell to an antigen-presenting cell that can activate T cells. Antigen-presenting cells display three types of protein molecules on their surface that have a role in activating a T cell to become an effector cell. 1. MHC proteins, which present foreign antigen to the T cell receptor. 2. Co-stimulatory proteins which bind to complementary receptors on T cell surface, and 3. Cell-to-cell adhesion molecules which enable a T cell to bind to an antigen-presenting cell for long enough to become activated. Before discussing the role of MHC proteins in presenting antigen to T cells, we consider the functions of two major classes of T cells. Effector cytotoxic T cells induce infected target cells to kill themselves. Cytotoxic T cells provide protection against intercellular pathogens such as viruses and some bacteria and parasites that multiply in host cell cytoplasm, where they are sheltered from attack by antibodies. They provide this protection by killing the infected cell before the microbes can proliferate and escape from the infected cell to infect neighboring cells. Once a cytotoxic T cell has been activated by an antigen-presenting cell to become an effector cell, it can kill any target cell infected with the same pathogen. When the effector T cell recognizes a microbial antigen on the pathogen, on the surface of an infected target cell, it focuses its secretory apparatus on the target. We can observe this behavior by studying effector T cells bound to their targets when labeled with antitubulin antibodies. The T cell centrosome is seen to be oriented toward the point of contact with the target cell. Moreover, antibody labeling shows that talin and other proteins that help link cell surface receptors to cortical actin fibers are concentrated in the cortex of the T cell at the contact site. The aggregation of T cell receptors at the contact site apparently leads to a local alteration in the actin filaments in the cell cortex. A microtubule-dependent mechanism then moves the centrosome and its associated Golgi apparatus toward the contact site, focusing the killing machinery on the target cell. A similar cytoskeletal polarization is seen when an effector helper T cell interacts functionally within a target cell. Once bound to its target cell, a cytotoxic T cell can employ at least two strategies to kill the target, both of which operate by inducing the target cell to kill itself by undergoing apoptosis. In killing an infected target cell, the cytotoxic T cell releases a pore-forming protein called perforin, which is homologous to the complement component C9 and polymerizes in the target cell plasma membrane to form transmembrane channels. Perforin is stored in secretory vesicles of the cytotoxic T cell and is released by local exocytosis at the point of contact with the target cell. The secretory vesicles also contain serine proteases, which are thought to enter the target cell cytosol through the perforin channels. One of the proteases called granzyme B cleaves and thereby activates one or more members of the caspase family of proteases that mediate apoptosis. These caspases then activate other caspases, producing a proteolytic cascade that helps kill the cell. Mice in which the perforin gene is inactivated cannot generate microbe-specific cytotoxic T cells and show increased susceptibility to certain viral and intracellular bacterial infections. In the second killing strategy, the cytotoxic T cell also activates a death-inducing caspase cascade in the target cell, but does it less directly. A homotrimeric protein on the cytotoxic T cell called FAS ligand binds to the transmembrane receptor proteins on the target cells called FAS. The binding alters the FAS proteins so that their clustered cytosolic tails recruit procaspase 8 into the complex via an adapter protein. The recruited procaspase 8 molecules cross-cleave and activate each other to begin the caspase cascade that leads to apoptosis. Cytotoxic T cells apparently use this killing strategy to help contain an immune response once it is well underway by killing excessive effector lymphocytes, especially effector T cells. If the gene encoding either FAS or FAS ligand is inactivated by mutation, effector lymphocytes accumulate in vast numbers in the spleen and lymph nodes, which become enormously enlarged. Effector helper T cells activate macrophages, B cells, and cytotoxic T cells. In contrast to cytotoxic T cells, helper T cells are crucial for defense against both extracellular and intracellular pathogens. They help stimulate B cells to make antibodies that help inactivate or eliminate extracellular pathogens and their toxic products. They activate macrophages to destroy any intracellular pathogen multiplying within the macrophage's phagosomes, and they help activate cytotoxic T cells to kill infected target cells. Once a helper T cell has been activated by an antigen-presenting cell to become an effector cell, it can then help activate other cells. It does this both by secreting a variety of cytokines and by displaying co-stimulatory proteins on its surface. When activated by an antigen-presenting cell, a naive helper T cell can differentiate into either of two distinct types of effector helper cells called Th1 and Th2. Th1 cells mainly help activate macrophages and cytotoxic T cells, whereas Th2 cells help activate B cells. As we discuss later, the nature of the invading pathogen and the types of innate immune responses it elicits largely determine which type of helper T cell develops. This in turn determines the nature of the adaptive immune responses mobilized to fight the invaders. Before discussing how helper T cells function to activate macrophages, cytotoxic T cells, or B cells, we need to consider the crucial role of MHC proteins in T cell responses. T cells recognize foreign peptides bound to MHC proteins. As discussed earlier, both cytotoxic T cells and helper T cells are initially activated in peripheral lymphoid organs by recognizing foreign antigen on the surface of an antigen-presenting cell, usually a dendritic cell. The antigen is in the form of peptide fragments that are generated by the degradation of foreign protein antigens inside the antigen-presenting cell. The recognition process depends on the presence in the antigen-presenting cell of MHC proteins which bind these fragments, carry them to the cell surface, and present them there, along with a co-stimulatory signal to the T cells. Once activated, effector T cells then recognize the same peptide, MHC complex, on the surface of the target cell they influence, which may be a B cell, a cytotoxic T cell, or an infected macrophage in the case of a helper T cell, or a virus-infected cell in the case of a cytotoxic T cell. MHC proteins are encoded by a large complex of genes called the major histocompatibility complex, MHC. There are two main structurally and functionally distinct classes of MHC proteins. Class 1 MHC proteins, which present foreign peptides to cytotoxic T cells, and Class 2 MHC proteins, which present foreign peptides to helper cells. Before examining the different mechanisms by which protein antigens are processed for display to the main classes of T cells, we must look more closely at the MHC proteins themselves, which have such an important role in T cell function. MHC proteins were identified in transplantation reactions before their functions were known. MHC proteins were initially identified as the main antigens recognized in transplantation reactions. When organ grafts are exchanged between adult individuals either of the same species, allografts, or of different species, xenografts, they are usually rejected. In the 1950s, skin grafting experiments between different strains of mice demonstrated that graft rejection is an adaptive immune response to the foreign antigens on the surface of the grafted cells. Rejection is mediated mainly by T cells, which react against genetically foreign versions of cell surface proteins called histocompatibility molecules, from the Greek word histos, meaning tissue. The MHC proteins encoded by the clustered genes of the major histocompatibility complex, MHC, are by far the most important of these. MHC proteins are expressed on the cells of all higher vertebrates. They were first demonstrated in mice, where they are called H2 antigens, histocompatibility 2 antigens. In humans, they are called HLA antigens, human leukocyte-associated antigens, because they were first demonstrated on leukocytes, white blood cells. Three remarkable properties of MHC proteins baffled immunologists for a long time. First, MHC proteins are overwhelmingly the preferred antigens recognized in T-cell mediated transplantation reactions. Second, an unusually large fraction of T cells are able to recognize foreign MHC proteins, whereas fewer than 0.001% of an individual T cell's response to a typical viral antigen, more than 0.1% of them respond to a single foreign MHC antigen. Third, some of the genes that encode for the MHC proteins are the most polymorphic known in higher vertebrates. That is, within a species, there is an extraordinarily large number of alleles, alternative forms of the same gene, present. In some cases, more than 200, without any one allele predominating. As each individual has at least 12 genes encoding MHC proteins, it is very rare for two unrelated individuals to have an identical set of MHC proteins. This makes it very difficult to match a donor and recipient for organ transplantation unless they are closely related. Of course, a vertebrate does not need to protect itself against invasion by foreign vertebrate cells, so the apparent obsession of T cells with foreign MHC proteins and the extreme polymorphism of these molecules were a great puzzle. The puzzle was solved only after it was discovered that 1. MHC proteins bind fragments of foreign proteins and display them on the surface of host cells for T cells to recognize, and 2. T cells respond to foreign MHC proteins in the same way they respond to self-MHC proteins that have foreign antigen bound to them. Class 1 and Class 2 MHC proteins are structurally similar heterodimers. Class 1 and Class 2 MHC proteins have very similar overall structures. They are both transmembrane heterodimers with extracellular N-terminus domains that bind antigen for presentation to T cells. Class 1 MHC proteins consist of a transmembrane alpha chain, which is encoded by a Class 1 MHC gene, and a small extracellular protein called beta-2 microglobulin. The beta-2 microglobulin does not span the membrane and is encoded by a gene that does not lie in the MHC gene cluster. The alpha chain is folded into 3 extracellular globular domains, alpha-1, alpha-2, alpha-3, and the alpha-3 domain and beta-2 microglobulin, which are closest to the membrane, are both similar to an Ig domain. The 2 N-terminal domains of the alpha chain, which are farthest from the membrane, contain the polymorphic, variable amino acids that are recognized by T cells in transplantation reactions. These domains bind a peptide and present it to cytotoxic T cells. Like Class 1 MHC proteins, Class 2 MHC proteins are heterodimers with 2 conserved Ig-like domains close to the membrane and 2 polymorphic variable N-terminal domains farthest from the membrane. In these proteins, however, both chains alpha and beta are encoded by genes within the MHC and both span the membrane. The 2 polymorphic domains bind a peptide and present it to helper T cells. The presence of Ig-like domains in Class 1 and Class 2 proteins suggests that MHC proteins and antibodies have a common evolutionary history. The locations of these genes that encode Class 1 and Class 2 MHC proteins in humans are shown, where we illustrate how an individual can make 6 types of Class 1 MHC proteins and more than 6 types of Class 2 proteins. In addition to the classic Class 1 MHC proteins, there are many non-classical Class 1 MHC proteins which form dimers with beta-2 microglobulin. These proteins are not polymorphic, but some of them present specific microbial antigens, including some lipids and glycolipids, to T cells. The functions of most of them, however, are unknown.